all to this meeting engaging with CSP, Neuberg Supratech Reference Laboratories about single cell genomics. So this is about today's meeting is about connecting with our certified service provider, Neuberg Supratech Reference Laboratories. My name is Dr. Sachin Minas. I'm the product manager at Premas Life Sciences. And Premas Life Sciences is the authorized distributor for many cutting edge biotechnology products, including TenX Genomics products. So we are very excited to bring you all together today and celebrate Neuberg Supratech's reference laboratory's achievement of becoming a 10x certified service provider for single cell analysis. This achievement means for you to gain access to cutting edge applications developed by 10x Genomics. So about Neuberg Supratech, this is not a new name. I think many in the biotechnology industry or the academia, they are already aware. Established in 1999, Neuberg Supratech is one of the most trusted names in pathology diagnostics today. And Supratech provides cost-effective, error-free, timely clinical laboratory services as well as research services, which we would be focusing on in today's meeting, specifically single cell genomics research services. And that's what they provide at international standards here in our country, in India. So once again, I'm happy to announce that they have become our partners now by becoming a certified service provider for our solution. And the idea of today's meeting is to build a sort of a community of researchers for single cell biology, provide latest information of new products and applications, then connect with peers and 10x and partners, and do all of this with our service provider, Neuberg Supratech. So this is the agenda for today's meeting, and it will begin with an introduction by our 10x Genomics India business manager, Dr. Amit Hussein, then followed by a short presentation on latest developments in single cell analysis presentation by our science and technology advisor from 10x Genomics, Dr. Yvonne Peterman. And finally, we will give the stage to Dr. Vishal Nanavati, who is a senior scientist at Neuberg Center for Genomic Medicine. So without any further delay, I would like to introduce our first speaker now, Dr. Amit Hussein, who is India business manager for 10x Genomics product. He is based from Bangalore, and Amit graduated with a PhD degree from Rajiv Gandhi University of Health Sciences in Bangalore in microbial sciences. And he has an extensive background in life sciences. He has worked in the life sciences industry for the past many years and he previously worked with Agilent and many other organizations on NGS and microarray technologies. So without any further delay, Avid, the stage is all yours.
Thank you, Dr. Sachin, for introduction. So let me share my screen. Can you stop sharing?
Yeah.
So hope you can see my screen.
Yes, Dr. Avid.
Yes, Doctor.
Yeah, thank you. So yeah, thank you once again, Sachin, for kind introduction. I'm very pleased to be here at the celebration of the TenX CSP event. Once again, congratulations, Dr. Vishal and your team for being certified as a first CSP in India. So please allow myself a few opening remarks leading into today's event, where we are highlighting the power of TenX Genomics single cell technologies. And you are also going to hear our CSP, that is Neuberg Supratech with Dr. Vishal talk. And he is going to share their experience and tips and tricks. And last one, but not the least, there is a most important announcement, that is a grant program. So I hope everyone will agree with me if I say 21st century is a century of biology. It aims to understand the mechanism of living cell and the increasingly complex hierarchy of cells and processes operating at the level of organism and even populations and ecosystems. So the biggest innovation of 21st century will be at the intersection of biology and technology. Right, the fusion of biology and technology that is blurring the lines between physical, digital, and biological domains. Consequently, the life sciences and healthcare market is growing, changing, and evolving. So our Chromium platform has been transformative for biological research that everyone knows, and our single cell gene expression and recently launched assay, that is gene expression flex, immune profiling, attack, and multi-omic assays have been enthusiastically adopted by scientists globally and have allowed you, our customers, to profile more than 5 billion cells. Recently, even the HCA published 3 large-scale single-cell studies in Science, totaling over 1 million cells spanning 33 organs, beautifully illustrating the power of these products to unreveal the complexity of biology. So the single cell revolution is only just starting. We are understanding human health and body at the single cell level, and even TenX Genomics tools are helping to uncover this. But we also need to understand the location of these cells. That is most important if you see in clinical perspective. So this was the concept for Visium. So using Visium spatial technology, that we released in 2019, we can be able to detect the genes and cells in a morphological context. But we also know that all these tissues are heterogeneous and very complex. So we are developing a new technology which is called in situ Xenium. So at TenX Genomics, we have developed and commercialized 3 complementary multi-omic platforms that we believe will become the ultimate toolkit for understanding the complexity of biology. So the first one, Chromium single cell. So this is based on microfluidic based chip and barcoding, and here we need to do NGS. So this platform has the ability to integrate whole transcriptome, epigenome, cell surface proteins, and more for large number of cells in large number of sample types, with different preservation methods to detect their true biological state. So this is perfect platform for hypothesis generation. So coming to the second one, our Visium spatial platform is a tissue-based 2-dimensional RNA barcoding solution with multicellular resolution. Also, we need a NGS readout. So this platform enables spatial whole transcriptome interrogation of cell types and states while maintaining the option to perform morphological analysis of the same section, bridging the gap between histology and genomics. So these 2 platforms are perfect platforms for hypothesis generation. And coming to the final one, the Xenium in situ platform is a solution for tissue-based in situ targeted RNA detection at subcellular resolution. So with an integrated microscopy readout, we don't require NGS sequencing here. So that means detection of mRNA species directly in the tissues itself with subcellular resolution, which is pretty mind-blowing. This is our perfect tool for hypothesis testing coming from your whole transcriptome exploration generated on the Chromium single cell and our Visium platforms. At TenX, we are constantly working on overcoming challenges. Importantly, we focus on not just individual assays, but entire end-to-end platforms. So when researchers invest in our platforms, they know that there will be continuing development and addition of new assays and features. This is perhaps best illustrated by our Chromium single cell platform, where since we launched in 2016, we have continued to innovate, release new versions of the existing products, and add additional products and capabilities. And we will continue to do this in the future as well. So the single cell gene expression part, Ivan is going to cover more on this, so I don't stress much, but I want to introduce you our latest additions to our portfolio. That is our new nuclei isolation kit. It is an easy use workflow to allow you to overcome the upstream sample prep challenges associated with working with the tissues. Especially you see when you isolate nuclei, you see a lot of debris. So this will hamper the results. So this is where this kit going to help you. And our gene expression flex kit, this is actually the best kit what we launched since we hear all the challenges, bottlenecks from researchers. So this uses a new chemistry to create our highest sensitivity single cell gene expression product and help to mitigate especially the storage, transportation, and per sample cost, all related challenges. So the other one, VCM Site Assist instrument, allowing easier access to FFPE samples and can work with even pre-section slides. So we don't, it is compatible with the fresh frozen tissue, FFPE tissue, even the pre-section slides. Just one slide is enough to understand the complexity of the data. And lastly, our new kit on the block, our Xenium in situ instrument, which helps to identify hundreds of genes and proteins simultaneously at subcellular resolution without NGS sequencing. So you can load one of these 5 assays onto the disposable microfluidic chips depending on your research goal. With these first 2 assays, single cell gene expression, you can choose to measure gene expression, plus a range of additional analytes like cell surface proteins and even CRISPR screening. So the 5' immune profiling assay lets you also assess the T cell and B cell receptors as well as antigen detection as an add-on module. So the single cell attack and multiome kits, our epigenetic solution is measuring accessible chromatin in nuclei. So our newest single cell kit is really revolutionary. Single cell gene expression analysis, it is becoming the new gold standard and brings you great benefits. Flexibility, used for FFPE fixed cells, high sensitivity, and ability to run single cell experiments most cost effectively. So to summarize, in order to exponentially increase our understanding of biology, we need to study biological systems using as many angles as possible. To enable this, we have built our platform to seamlessly integrate with each other. The way we see the science we are trying to enable is shown here. While we focus on building several robust platforms, we understand that researchers want to analyze many different samples and a large number of these. And at the other end of the spectrum, you want to perform many different analyses as well as tailored to answer your experiment question. In addition, you want to use several platforms to get different perspectives on the same problem. This requires assays that are designed to complement each other. So overall, I can say TenX is one-stop shop for multiomics research. So with this, I will end my talk and look forward to hear other exciting talks. Thank you.
Thank you, Amit, for this wonderful talk. So we'll now have our second speaker, and it is my pleasure to introduce Dr. Yvonne Peterman, TenX Genomics Science and Technology Advisor, who is going to talk to us today about how single cell solutions can add new dimensions to your understanding of biology. So I would just like to tell you a little bit about Yvonne. She did her PhD in biology at the University of Freiburg in Germany. She has extensive background in the area of life science business. She has worked at all the big names in the life science industry, whether it be Lumina as a senior applied genomic specialist, and Kaizen as a business executive for applied testing, Thermo Fisher, and Life Technologies. All of these big names Yvonne has worked at, and she, as you can see, that she has a lot of experience in the area of life sciences, both in academia as well as the industry. So with that, I would like to welcome Yvonne and deliver her talk.
Thank you, Dr. Sachin, for the kind introduction. I'm really excited to be here to present to you today in a little bit more detail about our single cell gene expression solutions from TenX. And I'm just sharing my screen now. Hopefully that will work smoothly. Is this the notes view or the correct view?
It's a notes view.
Notes view still. Let me swap this. Better?
Yeah, good to go.
Awesome. Okay, thank you. Okay, so I'm going to use the next 30 minutes to tell you more about specifically our single cell offerings. And Arvid has very greatly introduced all the different platforms that we have, and I will, so that means I can just focus on the single cell platform today. So just briefly beginning, we all understand that biology is immensely complex, and we know that really the biology, to really understand it, of all the trillions of cells that are in our body, to understand what's transcribed, what proteins are expressed, etc. We really need to have the perfect or the right tools at our disposal to look at as many cells as possible, as deeply and detailed as possible. And that's exactly what we focus on at TenX Genomics, and we sort of aim to deliver our tools along these three key factors here. And resolution obviously means, in our case, single cell resolution or single cell scale, but it also expands on that to mean more dimensions or simultaneous measurements of different analytes in the same assay, or multi-omics, what we also call it, to really ultimately get a complex or complete biological context. Scale is another important scheme. It doesn't necessarily start there for everyone straight away. You don't have to start big, but what's important is that once you have a hypothesis, or once you have a pilot study performed, you can then scale up, and scan and study more cells, more tissues, and more samples. And that's what our technology and the platform that you now have access to with this certified service provider here allows you to do. You can do small, low throughput kits, and then you can scale up and profile more. And this is exactly what we're here to celebrate today. It's the access. So now with this laboratory here, you have access to this technology, and access not only to the technology, but most importantly to the expertise that the team here has to help you really understand what the technology can deliver, and how to get your sample prepared to get successful, or to be successful with your data. So we've just seen this, our 3 complementary platforms, and for the purpose of today, we want to focus on the Chromium single cell platform specifically that we have in the market that generates and profiles all of the cells in your sample that you're preparing, and looks basically at the heterogeneity of the sample. So looking at this exactly is what are the benefits, or what benefits does single cell sequencing actually give us if we compare this? And if we start by using or looking at the bulk RNA sequencing, which is, what not that long ago everyone was doing, and it's still a very successful tool to compare, a healthy and a diseased person. But what we are doing essentially is we are averaging the gene expression signal from all of the cells that were within our tissue. So we don't really have an understanding of the amount of cells and the expression of the transcripts where they're actually derived from. And when we if we were to profile the exact same sample now with the single cell technology, we would be able to look at each subpopulation of the cell in that heterogeneous sample, and we would understand exactly which cell types are expressing which genes. And you can see with this illustration here that this can be vastly different. And as we understand and know or begin to understand, is that the smallest amount of transcripts of the smallest number of cells within your sample can actually have and drive disease progression, for example. Now, how we show these data, and most of you will be aware of this, I'm just including this here for completeness purposes. We are showcasing these single cell data in these plots, T-SNE plot or UMAP plot. So they're basically T-distributed stochastic neighbor embedded methods that here really the key is that every single dot represents an individual cell, and they're grouped by their overall gene expression profile. And they are color coded. You can actually change the color coding as well. But the key is that every cell that's grouped relatively closely together is quite similar in their gene expression profile. And when these dots are quite distant to each other, that indicates that their expression profile is quite different, and hence, or most likely, a different cell type. And you can see that each cell type has a very distinctive expression profile. And annotating, you do this by using marker expression genes that are described to actually map that back to specific cell lines, cell types. Now, a real-life example here, we are showing this is done with a lot more cells. This is actually millions of cells that this group here has profiled. And the authors here use the TenX technology to discern distinct cell types in the respiratory system and peripheral blood of COVID-19 patients, as well as healthy controls, to really better understand the impact of the COVID-19 viruses on the cell state. So to really, by unfolding that heterogeneity of the sample of every single cell that's present, we can answer many biological questions. So our solutions can help unlock answers to the cell types that are present, the lineages, the function of the cells, and you will be able to better understand of how the cells contribute to tissue and organ biology, hitting on all modalities that contribute to successful target identification, validation, preclinical and clinical studies, for example, in cancer. So some of the applications for single-cell gene expression include really elucidating the mechanisms of cellular development, identifying rare cells, rare cell types and states, developmental origins. People perform cell atlasing and as we mentioned, assessing tumor heterogeneity, detecting subpopulations of cells, and really drive the deeper understanding of biological processes. So looking at our Chromium single cell platform in a little bit more detail, really, we can profile tens of thousands or millions of reactions in parallel, as Amit introduced in the beginning. It's microfluidic based technology where we begin with your sample. You start with your sample that's either single cells or nuclei. That part is actually where a lot of the thinking and a lot of the expertise will go into to actually prepare that highly clean, viable, and debris-free cell or nuclear suspension to actually enable you to get really high-quality data out of the system. And this is where also your CSP laboratory here will really be coming in and helping you understand all these components. And so will also the Primus team or the TenX team to help you get this cell and nuclear suspension prepared. So within the system, you then partition all of the individual cells into individual nanoliter droplets. And following on from this process, you generate, by using the associated consumables, including gel beads and microfluidic chips, etc., you're producing libraries that will be sequenced on a short read sequencer, and after sequencing you will analyze and visualize the data utilizing our Cell Ranger software tool for single cell analysis and for interacting with the data Loop browser. They're both free. They can both be downloaded from our website. Loop browser runs on your laptops. If you haven't done so, you can go to our website into the support part of the website and actually download a large number of example data sets into the Loop browser directly and just play with the data sets and see really what the single cell data reveals. So looking into this a little bit closer into the reaction on the Chromium platform, really, the key here really is the fact that we have millions of parallel reactions. We are using these barcoded gel beads on one hand, and these gel beads are characteristic. They do have individual barcodes on them, and you load the cells and your enzyme and on the interface, and that all happens on the chip where the barcode and the cells and enzyme meet the oil. We do form these nanoliter droplets and incorporate the gel beads with the individual cells or nuclei. Now, the majority of these gems, how we call them, gel bead emulsions, will actually be empty, but then a subset of them will contain your individual cell or nuclei. And then what happens next, once we have actually performed the partitioning, we will then lyse the cells, make all the transcripts accessible. They will be captured on the gel beads, and then we perform a reverse transcription step. And in this reverse transcription step, every transcript that gets captured becomes or gets a barcode, a 10x barcode, and each individual transcript is also labeled with a unique molecular identifier. Then we can actually pull the samples, remove the oil, and prepare the sequencing libraries. Now, we've seen this slide from Arvid before as well. And with this slide, I just want to highlight that you can load one of these 5 assays onto the Chromium platform. So what you see here is the Chromium iX or X instrument. And really the first 2 assays here are our gene expression solution. And they are 2 distinct assays. The first assay captures 3' directly 3' transcripts, whereas the single cell gene expression flex assay, which is our newest assay in the assay family on the Chromium platform, is actually probe-based and specific for human and mouse. And I'll tell you in a minute what's so cool about this kit and why we think it will really transform how people conduct single cell experiments. And then with the next kit here. This is really when you are looking at immunology, when you're looking at trying to interrogate really all the immune components of a tumor, for example, or in general, any of the immune cells that are being attracted or that are being found in the tissues that you are studying, or in the model systems you are studying. The immune profiling solution, our 5' immune profiling solution, really is the tool here to use. And then the last two assays are our epigenetic solution. So we are really interrogating here the open or accessible chromatin with our ATAC-seq assay, or the multiome kit really allows you to measure both, to measure the accessible chromatin as well as gene expression. But I will go through all of these in a little bit more detail so you understand what all the different analytes are that we can capture with each of these technology, with each of these kits. And I want to begin with the 3' gene expression kit, because that's one of the kits that Neuberg is going to offer for you as a service. The key analyte really that you're looking at, as I mentioned, is the 3' gene expression. So you're looking here at any mRNA, any poly-A RNA molecules in your sample that is species agnostic, so you can really use any, well, almost any organism here to study this, and you obtain this readout of gene expression level on top as the basic readout, excuse me. And then with pretty much all our assays, you can add additional analytes on top of it. So that means with the same sample in the same run, you can analyze more readouts. So one, for example, is protein, so cell surface protein, for example, and because they are oligonucleotide labeled for detection, so not fluorescently labeled proteins or antibodies, we really take away the limitations that FACS sorting, for example, has, and allow larger proteins to be screened. You can also perform CRISPR perturbation screening at the same time as measuring, the actual gene expression influence that this has. Now, using this kit, what kind of data can you obtain? What's the sort of the level of resolution that you can get? And here you can see an example of just over 8,000 cells that we've analyzed from a PBMC sample. And you can see we are detecting really here a large range of immune cells that are being detected. So, we can see T cells that could be targets for immunotherapy in the future. We can see tissue cells versus tumor cells, and that really can help you identify biomarkers and drug targets that are expressed or present in tumor cells, but absent in normal cells. And you can see here also the epithelial cells, which line the blood vessels to supply nutrients and oxygen to the cancer cells. So in this data here, we also observed, or you can also observe a clear differentiation of B cells into plasma cells, suggesting a really robust B cell response in the tumors. So it's really interesting that also at this level of around 8,000 cells, you see a high number of T cells, but we can actually also identify the subpopulation of these affected T cells. So Th1, Th2, Th17 can all be identified just simply by using this 3' gene expression kit. And really the key message here is when you do this, this can actually not be seen if you just simply do FACS analysis or bulk RNA sequencing. These things would be masked, but can be revealed by using single cell sequencing. Here is another example where the 3' gene expression assay has been used to look at the onset, or to understand the onset of therapy resistance in small cell lung cancer. So the authors here, Stewart et al. From the University of Texas, they have really looked at the change in intratumoral heterogeneity in small cell lung cancer after platinum-based treatment. And this type of cancer is known for having a robust response to first-line platinum treatment, followed by relapse. So where less than 20% of cancers respond to approved treatments. And the median survival rate for these patients is also quite low, with just 12 months. So here the team really looked at circulating tumor cells from the patient, as well as using these to make xenograft models, and then developed single cell libraries from both these, using this our Chromium single cell 3' assay. And what they found was that there is an increase in intratumoral heterogeneity following treatment resistance. So at the top of the figure here, we can see on the right, on the top right there, we have 3 CTCs-derived xenografts from before treatment that are platinum sensitive, and you can see on the t-SNE plots that the cells are only generating 2 to 3 clusters per sample. And on the bottom, the model where the platinum resistance after treatment is shown displays between 4 to 9 single cell clusters. So really what that shows the authors is that there is a very small window of therapeutic vulnerability, and that is really important to understand, how you can improve these treatments. But this brings me to one of the key steps when you do a single cell gene expression or a single cell assay. And one of the key steps is, how do you get your samples? Who collects them at what time? And how do I preserve that sample? And how do I get it to the lab that actually is going to process the sample for me? And while TenX has many different types of protocols, validated protocols that help with these different types of things, there is still an overall problem when you can't actually have much of influence of how you collect your sample. And there's always the chance, if it's not done right, that it affects the biology of your sample. And this is where we've made a tremendous improvement in our assay offering with this new assay that we've just released, which is our single cell gene expression flex assay. And it is really becoming the new gold standard. And the key here really is, and I just want to spend the next few minutes explaining this kit to you, is it can actually fix and store. You can fix and store whole tissues or suspensions of cells and nuclei in 4% formaldehyde at the time of collection. It's a very quick process. You need our sample prep kit for that, and you need paraformaldehyde, that specific solution. But once you've fixed it, you can store your sample safely for up to 3 months and keep collecting samples until you have enough samples to run your trial or your samples in a very cost-effective way. The key here is this fixation method preserves your biological states. It preserves your fragile cell types right at the point of collection. We have inbuilt, and I mentioned this earlier, this is a probe-based solution. So it's a large probe pool targeting about 18,000 genes in the human and mouse genome. Into these probe sets, we have built in barcodes to facilitate multiplexing. What that allows you to do is you can actually scale up your experiment. You can pool up to 16 samples together and then run that in just one individual lane on a microfluidic chip. So that is up to 128,000 cells per individual channel, which still means when you pool 16 together, you can run 8,000 profile, 8,000 cells per sample. Because these probe sets that we've developed are really highly specific and sensitive, we used a tiling approach here to cover all the different genes. We have been able to reduce the amount of sequencing as well that's required to profile your cells by half. So really there's another saving that also comes in for you. And very excitedly, we have actually data as well that shows we can use this kit on dissociated FFPE tissue. So just very briefly, the actual workflow is not all that different. The difference is your fixation and permeabilization and storage, and then each individual sample, when you are ready to process them, will be hybridizing the probe set individually of each sample before you pool and then put that through the gene expression workflow. A quick look here at our probe design. There are really two sides, two hands to them. They both have to bind to their target site very specifically, and only if that occurs with basically a perfect match, the ligation will occur, and that's actually what we measure. We actually capture that probe. But what I want to put your attention to is this part here, which is that probe barcode that I mentioned that's actually inbuilt, facilitating the pooling. And something that we see, and we've first shown this internally, and we are seeing this now done by our customers that are using this kit. This kit is actually really even more sensitive than our existing 3' gene expression kit. And really here, we are showcasing that even if you use the same sample and split it into 16 different pools, there is really no batch effect. It really is almost identical, the profiling, and you can choose to do that obviously for 16 different samples, but you can also pick and choose if you wanted to use several barcodes for the same cell to actually profile more cells. And again, on the right here, I want to point out that you only need 10,000 read pairs per cell to profile your sample. Now, something that I'm personally really excited about is that this kit also allows you to perform single-cell gene expression of FFPE blocks. What that means is you actually use about 2 to 25 micrometers of an FFPE scroll from a tissue, and then following our protocol, you actually dissociate that following sort of a mix of an enzymatic digestion and a mechanical digestion, and then you can put this through our gene expression flex workflow. And here's an example of what this looks like with an invasive ductal carcinoma FFPE block scroll. It's actually fascinating to see how many UMIs and genes per cell we are still detecting, well over 1,000. And also when you look at the different cell types that have been identified and how nicely they actually cluster. So really exciting. And I just want to briefly brush over this here, over this capability. And I want to guide you to this protocol that would enable you to do this with the barcode, but we will also provide the information for you later. So really this kit does have a lot of advantages that come, sensitivity, the flexibility mainly for you to actually run your sample and store your sample and fix them, etc. So really an exciting new product that we've added to the family of products. Now, with the TenX Genomics assay, or with the chromium single cell instrument, you can also really, when you go into immunology, profile deeply with the immune profiling solution. Now, I know that's not necessarily one of the key focuses of the CSP lab here. I just want to quickly introduce it to the audience of some of the key questions that you can answer by utilizing the specific kits. You can see how the immune system responds to infection, how pathogens modulate the host immune system, and what the immune response, for example, is to a vaccine or a drug that's been developed. We know that the immune cell or the immune system is really diverse, and the immune cell diversity and antigen receptor diversity is really what drives that. And to dive into this and to capture this information, we really need to see the full-length VDJ sequences, because that ultimately forms the surface immune globulin B cell receptor light and heavy chains, and the T cell receptor alpha and beta chains. And that's actually exactly what our kit does. So at first you capture your digital gene expression. We are doing this from the 5' end with this kit specifically, because that allows you to get that full length VDJ information. And if you want to look at the T and B cell receptor information specifically, what you do is actually you perform a specific enrichment PCR step from the whole transcriptome mRNA molecules that were captured to really pull out these VDJ sequences. So it's basically just an add-on step that's performed on the libraries. And again, with this kit, you also have the option of protein and CRISPR screening, but I want to highlight one thing, which is antigen recognition for immune receptor mapping. So that's something that we enable now as well, where you can screen the binding specificities of multiple antigens or epitopes for hundreds or thousands of B and T cells, also with our latest edition, our Beam kit. And this here is just at high level what this data would look like. So with the 5' gene expression, you can still pull out all the different types of immune cells. You can cluster and characterize them, but you can also zoom in, for example, in this B cell cluster here, the brown cluster in the middle, look in, zoom in and see the clonotypes specifically, and you can do the same with the T cell cluster, for example, zoom in and pull out that TCR clonotype information. So there's a few really fascinating examples out there where this kit is utilized to really understand disease severity and response to therapy, here in this example on active multiple sclerosis. So the authors here have done or have tried to understand this disease severity and response by identifying expanded T cell or B cell receptor clonotypes. And they've used paired cerebral spinal fluid and PBMCs from subjects with relapsing remitting multiple sclerosis. And why single cell, you might ask, and the authors have stated that they've used microarray and bulk RNA sequencing previously to study B cells from these multiple sclerosis subjects, but have not been able to clearly identify or define the transcriptome-wide profiles of central nervous system B cell subpopulations, or were able to compare them with their peripheral counterparts. So this single cell resolution here really added a tremendous value in understanding this type of disease. And where else can the immune profiling solution come in? You can use this here to advance cell therapies, to discover targets, or to characterize an infusion product, specifically when it comes to CAR T. You can monitor the response of patients with CAR T cell treatment, or you can study resistance profiling. So we have examples in the literature for all of these applications already as a guide for you. And the last few minutes I want to use to talk about our last set of kits, which interrogates the epigenome. And really, when we look at it, ultimately what genes are transcribed into is the result of upstream regulatory mechanisms. So it's all great to look at gene expression, but to really understand why it changes and what changes, it's important to actually look at on the epigenetic upstream level. And this is sort of where our epigenetic solution or our attack assay comes in. And when you screen or when you interrogate the epigenome, what you can really learn or understand is the cellular diversity. Despite nearly identical DNA sequences, we can enable studying complex gene regulatory networks, because we can look at these drivers of differential gene expression across cells, tissues, and disease by identifying and looking at these transcription factors. And we can also characterize developmental trajectory or cellular responses to stimuli or drugs. So just quickly at high level, what we are actually measuring here, we have two kits. We have the attack and the Martion kit. So ATAC stands for Assay for Transposase Accessible Chromatin. So really the enzyme cuts anything that's accessible, the chromatin that's accessible, and adds a tag to it. So we can measure these DNA snippets of the active chromatin or the open chromatin. And that lets us really study the structure of the genome by identifies these regions that are open and active for gene transcription. And if we use the multiome kit and we add the 3' gene expression on top of it, we can see that by understanding what's open and active, but what does it actually relate to in the form of gene expression, if we can interrogate that of the exact same cell or nuclei, then we don't need to do inferred linkage analysis. We can actually read it out from the same cell. So these two kits really important tools to understand gene regulatory networks and cell state. And it's also on the menu, and I'm sure we will hear about this in a minute in more detail, what's on offer. And really, you get your gene expression clustering on with these data outputs, gene expression versus ATAC, and you identify much less cells, you might think, if you just use your ATAC data, but because you're putting this together and you're looking at it in conjunction, you actually get a much more defined understanding of these different cell types. And lastly, I just want to highlight that we have developed a kit, and we've released our first sample prep kit, which is the chromium nuclei isolation kit. And I'm bringing this up here because the multiome and the ATAC solutions, they require you to input nuclei, so nuclei only. And if you already have an established method in your lab for extracting really good quality nuclei, please continue using that. But if you're starting and if you don't have a protocol, it's worth talking to us to establish whether, you know, your tissue and sample type is suitable to use this, you know, a benchtop kit to extract nuclei. Just uses a centrifuge and about an hour of your time, and you can do about 8 samples at a time. So we have tested a lot of tissues that are compatible. There is some that are incompatible and some that may require some validation. So please, if you do want to use this kit, also if you want to use nuclei for any of the other assays that we have, just get in touch with us so we can advise you on the suitability. So with that, I want to wrap up my presentation around the chromium single cell solutions, the different kits that we have. And as Arvid also introduced, it's really talking about discovery mainly. The focused specific analysis goes more into the Visium and Xenium. However, I just want to mention that you can also interrogate specific targeted genes. So you can also use targeted panels in our chromium single cell assays rather than looking at the whole transcriptome. So you have both of these options to customize a panel and apply that to your sample if you prefer, rather than the whole transcriptome. There is a variety of support resources available that we can also point you to, and we are all available here to talk, but most importantly, you now have access to this technology. And with that, I finish my presentation. Thank you very much.
Thank you, Yvonne, for this wonderful presentation and insights. So, if anyone has any question, I would request you to type it in the Q&A box, and we'll take all the questions together at the end. And as I spoke in the beginning of this event, that this event is about celebrating Neuberg Supratex Association with us, and we now have our third and last speaker from Neuberg Supratech itself. I am very excited to introduce Dr. Vishal Nanavati to you. He's a senior scientist, research products at the Neuberg Center for Genomics Medicine. Vishal did his MSc in biotechnology from Department of Biotechnology at Gujarat University, and then he went on to did his PhD in regulatory biology from Cleveland State University, Ohio, in USA. After that, he also did two postdocs, one from Cleveland, Ohio itself, and another from Cleveland Clinic Foundation in the same city. So after that, Dr. Vishal joined the Neuberg Center for Genomic Medicine as senior scientist, and he has been there since April 2020. So Dr. Vishal, I welcome you. We look forward to your presentation about designing your first single cell assays using TenX Genomics platform. Vishal, the platform is all yours.
Hello. Thank you, Sachin. Thank you very much for this kind introduction. I really like to thank, first of all, Mr. Avid. I think we met pretty much almost like 1.5 years back, and when we planned that we would dwell into a site where all the Indian scientists and clinicians can come forward to learn more about single cell technologies. And thank you, Yvonne, for spending such a late night talk. I mean, I would like to let all my audience know that she's talking from Australia and it's almost like 11 p.m. there. So thank you very much. I'm going to share my slide now. Okay, is it visible? Yeah.
Yeah.
Okay, perfect.
Yeah.
All right. So once again, welcome you all. So the idea of today's talk was not to keep too much on the technical details, but rather giving you a sweet orientation of what Neuberg Center for Genomic Medicine is a part of Neuberg Supratech Reference Laboratories, what is TenX Genomics, and how Primas helps us overall in our regular functioning as well. So my name is Vishal Nanavati. I did my MSc, as Sachin said, from Gujarat University. I'm a local guy, went to US to do my PhD and postdoc there. Been lucky enough to be able to learn my genomics on multiple different instruments, including TenX Genomics. So my training on TenX Genomics happened in 2018 in my US lab, and we have recently a publication coming out of it as well. So I'm very happy that even after I left my association with them, but I'll be getting a publication out of that, which includes 3' UTR-seq, or more like a single cell gene expression, as well as the now with the Visium data set as well. Here in the right side, that's the barcode for the company, or like a more like a dedicated department of Neuberg Supratech NCGM Global. And I'll give you a little bit brief about the company, and then we'll guide you through the 3 different assays that we have performed by ourselves, helping a couple of clients. And we'll talk about pros and cons and what we have learned while we walk through this process as well. So number one thing, we are a certified service providers, and we are here to help you. We are here to help you for everything which starts from experimental design. Feel free to talk to me or anybody, even in Primas or TenX team. We can really help you out how to think about your experiment, right? And that's the most essential part. Of course, we all read a lot of science in general, right? We go through a number of publications. But it's really essential to think through an entire experiment as well as how the data is going to come out before you think further about it. So I think that's the very first step I would advise every listener, that even if you plan to do any kind of experiment, you have to work on your experimental design for it to work completely. The second one is that our presence, Neuberg Supratech's presence is in Pan India. We pretty much can collect samples from anywhere, which includes we support you for your sample collection and preparations for the single cell genomics as well, which can include tissues, PBMCs, cells, and nuclei or whatever. Once we discuss the entire project with you, we can guide you how to collect the sample. We can prepare even your collected samples if it is a tissue or PBMCs. And the site is prepped for that. We have been trained for that. The next one is that we can evaluate your sample. If you have isolated your cells, tissues, or nuclei, we can help you evaluate the sample. We can help you run it or run it on our in-house Chromium iX controller, right? So that's the Chromium controller what we have. We can prepare the final libraries. We can provide you all the QC steps. We can guide you through those. And I have the slides that is going to show you how the QCs looks like. We run it on our Illumina platform sequencer, which we have it here in our Ahmedabad facilities, NovaSeq 6000 and NextSeq 2000. I must add that the NovaSeq 6000 here is, we have it since October 2019. NextSeq 2000 we have since August 2021. So we are very seasoned on utilizing the machine. We can get the data out as we plan for. So we are very thorough with the techniques of utilizing our sequencers, and we can help you out with that as well. And finally, the most essential part that everybody would like to dwell into it is basically run the analysis. We can run the analysis on our own server, which we are trained to do the TenX provided tools such as Cell Ranger and Loop browser files. And we can run a couple of third-party tools as well. And we do not shy off of learning new tools if the client's needs are there. So that's the CSP site for you. That is the Neuberg Center for Genomic Medicine. This is a 3' gene expression services, which includes multiple different subsegments of that. And we are trained for that. We are here to help again. Just remember that one thing. And meanwhile, I would really love to see multiple questions bouncing back to me. So if you have any questions, feel free to raise. I think I wouldn't be bugged at all right now, or even in somewhere in the end as well. So that's our certificate. That's the nice certificate we get once you have the CSP site inspection and the assays done. This was delivered to us by Amit and the Primas team to us. That's our certificate, which is available on TenX Genomics website as well. You can go and search for us via CSP site across the world, and then you can find us there as well. And so that's our certificate. And we are very happy about it. We are very proud about it. The path to reach this is not a straightforward path. It was not a straightforward path. It took us about 7-8 months. So I wouldn't lie on that. And at the heart of everything is basically the hard work of the team and the support from the TenX and Primas. So we are very proud to have this certificate. We treat it as one of our best publication of this department so far. So this is our facility. I'll introduce a little bit. I mean, we have very big facility. It's a 5-floor building, which is a genomics building, dedicated genomics building. But I'm going to showcase you things, the required things only specifically for the TenX part. And we will welcome anybody who's willing to come and visit us at any time point. We arrange the tours as well of the site for multiple clients as well. So we have biosafety laminar hoods that can take care of your sterile condition of the tissue processing. We have the dissociators for the tissue dissociation as well. We are routinely using it in our oncology segment in general. So we are very familiar on how to get the single cell suspension done on multiple different tissue types. It does not require us to do a brainstorming now that what kind of a tissue requires what kind of a protocol. They are all very well validated and standardized. That's our Countess 3, which is basically the one which helps you to say whether the experiment is a go or a repeat tissue or a repeat extraction. We count our cells. We can also count the cells via hemocytometer under microscope, but that's been Countess 3 is basically one-stop quick solution, which is what you require from single cell suspension to the library preparation. So next column is basically TenX Genomics Chromium iX. That's there. This machine is present here in Ahmedabad in the second floor of this our building. And we utilize this to prepare and pass through your specifically for the gem preparation of different tissue types, different cell types. And once we have prepared this, which passes through the Chromium iX platform, we have a final gems. We do a couple of QCs at the cDNA level, at the library level, and for that we have a tape station as well from the Agilent, which allows you to QC your final libraries before we pull it on our sequencers. The sequencer, as I mentioned, we have our in-house Illumina NovaSeq 6000 and Illumina NextSeq 2000. And once we have this data set, the data can be accessed from or processed from our in-house server that has been current capacity of the server is up somewhere around 300 terabyte of the space, with up to 1 terabyte of RAM and about 4 nodes. So that being said, that it can process somewhere around 5,000 to 6,000 sample per day. That's what it turns out to be, right? And that's what we are routinely working with. So the bulk is never a question. The workload is never a question for Neuberg Center for Genomic Medicine. We are being pretty much handling it very nicely. So with that, that's the facility so far. So this is what we have. And this is, you can see like, we have not taken any picture from anywhere. It's the raw pictures taken from our phones. And whenever you will walk into our facility, that's the machines you're going to see as it is. And that's the reason to keep it as raw as possible. So with this, I'll switch a gear a little bit about to start with the how to design about our first experiment. So your question would be that when I need to when I need this particular technology, right? That's the question you should ask. So the number one is like that bulk sequencing or bulk transcriptomic is something like a fruit smoothie, right? Where you mix multiple fruits, you crush them, and ultimately you enjoy a cold smoothie. That's what your bulk RNA-seq is as well, where you pretty much have a tissue or cell types. Think about it like you might be treating your samples or the cells with some sort of treatment such as RNAi or CRISPR knockout or CRISPR knock-ins, or certain sort of a dose dependency treatment of the pharmacogenomics, or the CRO people who might be collecting samples from the patients or the patient samples in general. Whatever you look into the NGS as of now, that's the one which is routinely done. You basically take whatever the biological material or fluid you have, you make a crush of it, and you present it the data in that form. Now, as far as that's informative, it loses out the information of each fruit that is present, right? If you are bad like me, who does not understand much pellet information, I would never be able to differentiate between the two fruit conditions, right? But if somebody is very good, they will be able to do it. Similarly, in a single cell scenario, when you have a single cell transcriptomic done, you can identify each cell's transcriptome, right? Or it is represent, or the way it is represented here in this picture, where you can clearly identify each different fruit, right? And then it retains basically the transcriptomic information or the flavor information when you're eating one fruit at a time, right? The third one, which is the part of the TenX Genomics platform, and we are not a CSP as of now, but we are planning to launch that pretty soon within next pretty much next financial year, and that is a single cell spatial analysis, or the way they are talking about in the VZM format. So this is more like a fruit cake, right? Like when you are taking a piece of it, you might be having that a scared present orange or not from that cake, right? So that's the information you can, that kind of an information, or that's the analogy we like to put it forward to when we are describing these technologies, right? So if your goal is to learn about the fruit smoothie, it's still a bulk sequencing. If your goal is to understand that you want to identify each gene's transcriptomics, right? That's when the single cell comes into the picture. And I guess if the format allows, I would like to take one of the question that has been asked is basically, which format or which platform is suited for rare autosomal recessive disease, right? I mean, Yvonne has responded to that without reading that much, but I would like to add there a little bit about it, right? So with rare autosomal diseases, you might be doing a whole exome sequencing or a genome sequencing, right? So you're only looking at the DNA part of the central dogma of life. What single cell genome technologies, or rather 10x genomics, is providing you the information is that the one stream down after the information of the single cell, the rare autosomal recessive disease information. You would be able to identify what particular cell type is behaving in a certain format based on the transcriptome, which you can compare with the, in general, the data bank which is available to us via multiple reports and data. And we can conclude from that what this particular genotype is giving you this kind of a phenotype if you are a clinician, and now this is the molecular baseline for that. So that definitely helps. So that is when that's how you decide whether you need a single cell platforms or not, or single cell technologies or not. With that, I'll move further. Okay, so how to choose the best experiment for myself, right? Like, that's the first question I would ask to myself as well, right? So if you go through a 10x Genomics support website, that's the first screen you see, right? That you see basically single cell gene expression, single cell gene expression flex, single cell attack, single cell immune profiling, single cell multiome attack plus gene expression. So out of all this 4 or 5, which is the one that is best suited for me, right? And so from here onwards, I'm going to go a little bit detailed one at a time. Yvonne has nicely put up all the information as you want. We are going to give you a little bit about our experiences around that as well. But each different assays is going to give you, for one thing is very essential. It is going to give you transcriptome information besides ATAC, but it also gives you the different cell population. So your number one goal becomes is that basically you are going to identify different cell populations. How to interpret that data is a question of an entire next talk. We cannot cover it within this time frame, but I'm more than happy to talk about that, how to interpret that data in the next talks or anytime soon. Right. So we'll start with the single cell gene expression. Now, once I click on single cell gene expression, there are other applications along with it as well, right? So you can identify cell surface protein. You can go through high throughput assay or low throughput assay, CRISPR screening, or cell multiplexing as well. So now this allows you to do a more detailed information, right? So if your assay is something like, let's say you are a CRO company who is working for a pharmaceutical company, right? And your goal is to identify PKPDs of the particular drug treatment, right? And you are sure that that particular drug is inhibiting a certain cell surface marker, but at the same time you want to query the information which is present in terms of transcriptomics in order to call for your PKPDs well. That's when you identify a cell surface protein and you utilize that particular assay, right? And then once you do a cell surface protein along with the feature barcode technologies and then perform a 3' gene expression assay, you not only identify a particular cell population with expressing a certain cell surface protein, but you also differentiate the gene expression present in that particular cell among with the other cells which you are going to capture during that process. Now, if you imagine that, that's the kind of a world you would love to work with, right? Because it's the amount of data, it gives you a certain kind of a clarity about your particular experiment or your particular molecule, which in a way may reduce the number of patients you have to recruit to do your, stage 2 or stage 3 type of a clinical trials. It reduces your clinical trial burden on the mouse sample. Right. So this is the technology to give you more information with a minimal number of samples, which allows you to manipulate a much more at the molecular level as well. And this goes through an NGS technology, which is pretty, which is pretty foolproof in a way. I would say that it provides you a certain definite information. The CRISPR screening is another experiment which you can think of, that if you want to do a CRISPR knockout of particular region in particular different cell type, let's say this is going to be in a blood sample, right? You're going to work with a melanoma kind of a condition, right? And you want to study a CRISPR screening on that a particular region. You can design a multiple targets. You can knock into the cell type, cell culture condition, and you can query each particular CRISPR target along with a different transcriptome information. You can decide whether the CRISPR targeting gene A is affecting gene XYZ in a certain manner, or CRISPR targeting B, which is affecting target XYZ, right? So this provides you an information not only with the one assay, because doing a single, or whoever has done the CRISPR assays, one knockout or one knock-in at a time, they know it requires a tons amount of energy. It takes about, I've done that by myself. It takes about 4 to 6 months just to get the correct cell lines. And then the data analysis takes another 2-3 months for that. Right. Compared to that, you can do a standard knockout or knock-ins in a single library. Within 3 months, you have a complete data set. It reduces your time, and time is money, which compensates for the in general, the applications of the 10x genomics and or in general, right? Cell multiplexing, again, it's a multiple ways where you can pull multiple different type of cells and run it together, which happens to be a cost-effective manner to identify different cell population. So now I'm going to talk to you about a single cell gene expression 3' assay, which was technically a CSP assay. We were required to do this assay. The conditions were set in a way that the 2 different technicians should perform this. There should be 2 different sets of PBMC cells, and we need to run it in a duplicates. And then, so that's what we have done. We needed to achieve a certain targets of the in terms of a sample QC, library QC, data QCs, and that's what I'm going to share with you. So we achieved the we collected the from the 2 healthy women. Okay, so these are completely healthy women, and we collected the blood sample from there, isolated PBMCs from them. We achieved the 90% of cell viability in them. The cell counting was performed using the machine which I've showed you. I'm sorry for the typo. It's count as 3, not 2, with the trypan blue staining. Okay, and that's the data set we have. This is the actual image we have taken. I'm sorry, it's not zoomed, but in general, you can see it has basically 97 and 93% of the live cell viability there, right? Between the 2 samples, the sample names are named as M and S in order to, by keeping the identity hidden. Once we finish that, we process the libraries using the protocol CG315. That's the way I would rather remember all the protocols. And that the protocol's name is Chromium NextGem single cell 3' gene expression, which was version 3.1. It's a dual index library. This is the image you get once the samples are processed from the single cell Chromium platform controller. Now, if you see the image in the right, that's a sample S of replicate 2, and a image in the left, which is sample M of replicate 2. Now you see the right one, the sample S has a perfect recovery of all the entire 100 microliter of gem, while we recovered only 98 microliters of the sample M. The reason I'm showing you this is that although we say everything is perfect and rosy, things happens, and we could still go ahead and run the data, and this kind of a scenario can happen as well. And we are trained to take decision on certain conditions as well while we are doing the QCs as well. So once we prepared the final libraries are sequenced on a SP100 flow cell on Illumina's Nova 6000 instrument, which was used as a sequencing on a paired end condition, which was suggested as 28, 10, 10, 90 parameters. Data was demultiplexed using 10x Genomics Cell Ranger ARC tool, and web summary and clue files were generated for data analysis that was submitted to 10x Genomics FASs. And that's when they validated our entire experiments and approved us for the single cell CSP site, right? Now, once we got that, I mean, being a scientist, I had this, itchiness in my body that we need to dive deep into this data set as well, because we wanted to utilize this data set. And this is where the beauty of this 10x genomics come into the picture, because remember, we have taken the blood from 4 so-called healthy women's. Okay, they are in their pretty much reproductive age, and there is no known clinical history behind it, but the data suggests something else. And that's the beauty about 10x genomics. And I'm very happy about that we decided to have this kind of a subject, because it worked out not only for us to do a CSP assay, but it has certain biological relevance among the humans, which is pointing out at that we might need to do a precision medicine very soon in the next decade or so. Right. So that's the data set we got from our web summaries. Like you have seen, the sample M and M2, they are pretty close enough in terms of number of cells we have collected, the data mean reads, and the number of genes we could capture out of it. Same for the S1 and S2. That's the heat map we have generated. That's from the clue file, which was collected from the Cell Ranger ARC. And you can see clearly that we have differentiated the generalized data in a 3 different markers. On the left, you can see B cell, T cell, and monocyte markers, right? For sample M1 and S1, and I've only selected the down-regulated genes. Now, if you see the region, the same region, right? Like you can see the down-regulated genes among the B cell marker, they are pretty unique and similar in both the samples. So this shows that among the healthy individuals, right, the pattern is pretty similar, right? The list of genes nicely overlap, but it also had some unique to sample M1 and sample S1 as well. And similarly for the monocytes as well, right? So this was the intriguing idea that it did not only had an overlap of the number of genes that are up and down, or rather down regulated, and there are certain unique genes that are very unique for set M and sample S, right? And we looked in further detail for that. So we generated, so that's the standard, you get a Disney plot. This is a Disney plot from the same two sample, and you can see the sample M1 and S1. Although we are calling them as a two healthy individual, identifies a different sets of B cell and T cell marker among with the monocytes, right? And when we looked at the CCR7, this is a chemokine receptor 7, which plays an essential role in immunity and tolerance, right? And we queried for that particularly among this set of cells, because that's what makes more sense to look into the immune markers. So you can see this particular cells or particular chemokine receptors expression is very high in T cell, as it should be in the sample M. But when you look into the sample S, right, the expression of CCR7 is quite high compared to the B cell of the sample M versus S. Although again, remember, this is the healthy controls, right? And you see a completely different conditions or different scenarios of CCR7 present. The chemokine expression is completely different. That question does that, okay, there is certainly a certain immunological conditions going behind this patient or the client is sample S, right? So what is that? That could be a different scenario. Now, my training at Cleveland Clinic was in an epigenetics, right? So I thought in that angle that what could be the idea of different DNA methylation markers or the standpoints, and that raises the concern that let me ask very first question, that what is the scenario of those, whether those DNA methyl transferases are even expressing in these cells or not. So when I looked for that particular set of genes which plays an essential role in DNA methylation manipulation, you can see I looked for DNMT3-1, DNMT1, and DNMT3A and B, and among TET as well. So this set of DNMTs are DNA methyl transferases, which adds the methylation on the 5th position of cytosine, and TET1 is an enzyme which removes the methylation from this cytosine, right? And you can clearly see that among M and S1, so M and S, there is a different conditions of the expression of this DNMTs and TET, which suggests that of course there is a completely different expression pattern, or the methylation, but there should be a completely different methylation pattern among this set of first of all sample, and among that sample it could be a different scenario of the in the methylation conditions of this different cell types, right? And that could lead you to understand the basic immunology among these patients or the clients as well. So not only that, for I have looked for tens and 20s of different proteins or rather genes expression among this set of sample, and all of them is pointing out at one thing, that sample S was undergoing a certain sort of immunological conditions while we collected the samples, right? So that's the data set we have. Now that raises a good question, right? That of if DNMT's conditions is different, methylation will be different. It is known from the literature that methylation can affect a different chromatin accessibility, and it would be nice to study chromatin accessibility. So that's where another segment of the TenX comes into the picture, or the rescue, that there is a single cell attack assay. We did not perform single cell attack assay on this particular to sample. We intend to do it at some time forward, but then this is our second assay that we have successfully finished. This is one of the clients samples. They provide tissue, and we utilize that. This is the protocol we utilize 496 to basically preparation of the library. We definitely utilize the Miltennium X to dissociate the single cell suspension. We utilize the basically yeah, so that's what we received. We achieved 92% of the cell viability from the tissues that we had. 4 of them, this is just a one representation, 4 of them were above 90%. And then we utilized the manual protocol to prepare a single cell nuclei suspension, because when you're performing single cell attack, you have to perform convert your cells into the live to the nuclei isolation. Now remember, between just if you pay attention between left and right, count as 3 images, you can see 92% live cells and about 90% dead cells. That is technically providing an idea that we could isolate the nuclei, because when you are performing a single cell nuclei isolation, trap and blue tends to trap and blue does not get absorbed, and that's when you get a more dead cells. That those are actually a true nuclei, which you can zoom in into the slides and identify the nuclei as well. So once we had this clean go, we prepared the library, and this is what the library should look like. I mean, you can have n number of representation available on 10x support system as well. And this is what we got, the tab station profile of the single cell final library representation is there. And this libraries were again sequenced on a special flow cell SP100 flow cell on Illumina Nova 6000 using 5, 8, 16, 90 parameters. That's the one which they are suggesting for the protocol, and the data was beautiful. Data was demultiplexed using Cell Ranger ARC tool, and again web summary and clue files are generated, and I'll showcase you a little bit of outcome of that. Again, this is one of the sample. We could basically estimated about 10,000 cells, roughly 9,447 of the high frequency fragments, and 54% of the fractions of the high quality fragments were overlapping the peaks only. So which was giving a good proof about the data set. That's the data set of the cell outcome. Like you can see the cells are slow, with a nice curve about the cells, about the QC. This is the insert size. That is one of the way to QC your single cell attack data set. And this is what the regions, which is showcasing that the transcription, the enrichment of the peaks, what we are identifying is nearly around the transcription start site. And that's what you would usually expect from the attack assay. Attack assay is basically going to look for the region which are open for the genome and not bound by the nucleomes. And that's what is turned out to be the scenario. Most probably your promoters tend to be open, or the region -500 to +500 tends to be open, right? And so out of 4 tissue, control tissue shows a classical increase in accessibility in the region of the genes involved with the higher expression in T helper cells, such as MAP kinase region. So again, from the loop file, we have done a little bit of secondary analysis with the permission of the client. And we identify that the loop, so this is the track. Again, we have differentiated among the 3 classical subtypes of monocyte, B cell and T helper cells. So this is the region on chromosome 17 on the MAP kinase region. So position number starting from 1695 million base pair to 17 million base pair is the MAP kinase region. Now, by looking at it on the left side, you see the units of the cluster of the peak size. It does not make sense from here onwards, but if you pay attention to those carefully, so this is the MAP kinase region and the left is the unit. You look at those peaks, right, among the monocyte, B cell and T helper cells, right? Just by visualizing to it, you would come to know immediately the different openings of this region, right? These are the regions which are completely different. It represents that, of course, not only that you can differentiate the cell cluster based on this type of phenotypes, right? But what it tells you that among the monocyte, B cell and T cells, there should be a different level of MAPK associated transcripts, right? Which plays an essential role in innate immunity, right? And that's when you expect that MAP kinase should be highly active in T helper cell, and that's what you are seeing in the overall increase in the accessibility of MAP kinase region in the T helper cells, right? And now, see, the thing is that if you have done this single cell attack assay, right, you get this nice opening region, right? And now you would want to know that what is the scenario of this transcript, right? And that we don't have it here, right? Because we did not perform a multi-omic assay. And that's when basically we need to start working on a multi-omic assay. So again, we have successfully finished a single cell attack assay in our in-house facility as well. So now I'm going to talk to you about one of the sample we have performed on a multi-omic assay, which stands for single cell attack plus gene expression assay. So the background history about this sample was that it was a FACS sorted T cells. And of course, there was a treatment behind it and were provided for the multi-omic project. The client visited site to observe and understand the quality requirement of the project, right? So it was not a straightforward work for us because even client had to work on to the storage and shipment conditions. So they came to and visited our lab, they sat with us, they saw our processes, they brought couple of cell types among them, they utilize our hoods to work around the conditions as well, right? So we really help them to understand their cell viability conditions as well, right? And similar process of cell counting and nuclei isolation was performed as single cell attack. Library was prepared using this CG338 protocol, which basically is the single cell multi-omic attack plus gene expression reagent using a dual index conditions. So, sorry. Yeah, and the final libraries are sequenced basically. Now, when you're preparing a single cell attack or rather multi-omic libraries, there are two different libraries at the end of the day or at the end of the final experiment. One of them is basically the library of gene expression, which you need to sequence on a 2810, 1090 chemistry, similar like 5' or 3' assays. And the single cell attack of the multiome has a little bit longer index, and then that's when you utilize a 58, 24, 50 chemistry. Again, that was done on a separate flow cell of SP100, right? And this is something that anything which needs to be sequenced, that I really suggest that you leave it on us, because we have done that over a number of times, right? So we have done that, we got the data, we demultiplex it, we passed it through cell range check, generated the loop file and web summary. So that's the web summary outcome. We collected roughly about 16,000 cells. I still feel that this is a little bit higher. There was a number of multiplates there. We could cover about roughly 2,521 ATAC regions and about 292 gene expression regions, right? Then there was a ATAC cluster. That's what we received as a ATAC cluster. And this is what we have seen as a gene expression cluster. Now you may ask that there is not a nice separation here, but remember, these are the sorted T cell after a certain treatment. You would expect a less heterogeneity among this. And that's what we are seeing here as well. We are identified different number of clusters based on ATAC and gene expression studies. We worked through that. We annotated the different clusters again in our standard scenario. But before that, let me go through the certain QCs. So this is kind of a textbook QC you get when you are doing a joint cell cluster analysis. This is a kind of a textbook QC you get when you do a ATAC peak clusters. Again, transcription start site opening and peak enrichment and the insert size distribution in the multi-omic ATAC scenario, right? And this is basically the data set. Again, these are in the 8 clusters, but if you think, or rather, let me explain you that the cluster 1, 2, and 3 are belonging from the B cell, 4 and 5 are from the T helper cell, 8, 7, and so 6, 7, and 8 are the monocytes. And then on the top, you have a gene expression cluster. In the bottom, you have ATAC clusters. So now from each different cluster, not only you have a different region opening information, but also the genes that are highly up and down regulated among those particular cell types as well, right? So if I just pay attention to only one cluster here, right? So on the top, when you're looking at the gene expression, these are the genes that are expressing differently. And then the bottom, you are seeing the regions that are differently behaving. We are currently working with the client to dive deeper on these different sets in order to come out with the beautiful analysis that they have worked with on. They have done this essays on that. It might give out a different, super enhancer or transcription factor based scenario in a different immune cell conditions, right? So that's the beauty of single cell attack, that not only in the region, but on also the gene expression we could identify. And that's what we have done successfully. We are currently working on that as well. So that we have successfully done that, and we are planning to do more assays on different conditions. So finally, a single cell immune assay as well. That we tried this assay. We are still putting it as an under validation in with the lab. We could not achieve the first few rounds of the assay of the sample submitted by the clients. We did not try it by ourselves. Uh, One of the most essential thing here, as I'm saying, that again, we are here to help. Talk to us openly, and we understand. And after going through an entire cycle of experiment, we realized the client did not provide it, or rather clarified the sample tissue of processing condition. It turned out that the tissues were frozen almost 2 years in an RNA letter. Now, if you think about it, if you're planning to do immune profiling, you need a cell surface marker well and healthy, and RNA letter tends to dry up your tissue. It of course protects the RNA information but does not protect the cell surface marker information, and that's when the experiment failed. We are trying to currently work through this essay as well. And while we are trying to work through that, currently company is planning to run a last quarter special discount for somebody who would like to help us and work with us and dive deeper into the further establishing this essay as well. And you can reach us out for further details for that. Now the final one, and Yvonne has gone into length for that, which is the single cell fixed RNA assay. It's a flex assay. Again, she has presented the same slide, so I'm going to skip for that. But I think this is one of the first, or rather the best suitable reagents for clients like us. We understand the market in India. We understand the geography of India. Pretty much every sample reached to us within 24 to 48 hour of time point. That could be a very crucial time point. At the same time, the funding dries up very quickly. So that is when the fixed RNA assay comes into the rescue, because we can help you either fix the tissues at your end, or we can help you out with the preserving your precious sample at our facility when you are running multiple different assays. Clubbing the more samples always going to benefit you. Again, if you think about it, that you are going to utilize a formaldehyde to preserve the biological state of the fragile cell type at the point of collection, and all you need is a fixation kit with along with the 4% formaldehyde. If you don't want to procure that, we can help you out with that as well. And you can plex as much as up to 16 samples per well, which brings down the price point by 16 times, right? Like if you think just a simple math, all the scientists can do that pretty quickly. So again, for the fixed RNA, this is what you need. If you're going to look for fixation at your facility, this is the reagent that you must have from the 10x before you start collecting your tissue or you plan to work on it. It is better to fix them fresh rather than waiting for the reagents to come. So again, plan your experiment correctly. And this is the region where the client must needs to identify and read through these protocols. So that's the assay. I must put up a disclaimer right now that it works well on human and mouse because that's the probe hybridization available, and 10x is working to develop further reagents for that. So again, finally, the India is basically an agricultural country. There is no doubt. India or Indian government spends more than 50% of their budget on agriculture segment. Recently, CRISPR modified or genetically modified plants segment is opened up. That means you need to have a plant segment available. We have one brave scientist working with us. He's been working with us reluctantly. I mean, he's been submitted us a sample which is almost like 6 to 7 times. So I would really appreciate his efforts and patience with us to help us establish this. So for plant, you instead of nuclei, it is called protoplast, and protoplast preparation is must to perform in order to perform a single cell gene expression. And we are currently suggesting clients to do that at their end first. And then not only is protoplast isolation, storage and thawing process as well before single cell gene expression can go on a 10x genomics platform. We have done this multiple times. We have reached pretty much, up to 72% of viability after 48 hours of travel of the tissue. So we are very hopeful that we will cross that 90% of live protoplast mark pretty soon. And the scientist is developing the protocol to, even ship it. And probably if that works out, I think we don't mind working out with any other scientist to help you out, to work with your plant single cell assays as well. And we are really expecting a major breakthrough coming through this in coming months for the plant biologist as well. So we are here to help you out with that as well. And you can utilize our services for the validation expertise as well. So with that, which brings me to the final part of my talk, and I'm very, very, very happy to announce a grant program. We are launching a grant program which is going to run from December 1 to January 2023. This is a CSP grant program which suggests basically, we invite all of you to apply for this grant. We are going to review the grant. The grants are going to be reviewed by the FASs at TenX, Premas, and by us as well. And we are going to announce one winner, and we are going to provide basically I think up to 4 reactions of it. I guess probably you might want to chip in a little bit. And we are going to help. We will be the site where we are going to work along with you, and we are going to provide you the results. So if you think this could be somewhere around up to 1 year of funding, right, for any scientist. So I'm very happy that we are launching, and it's available. It's open from today now onwards. Please scan through the barcode to reach out. We are going to come out and put it up in our social media sites as well. If you still have any concern or questions, you feel free to reach out to me at Neuberg, or Avid at TenX Genomics, or Sachin, or any of the FSs of the Primas team has, and we will be happy to guide you through the application process as well. With that, I have certain announcement from the NCGM site as well. So number one is basically, please participate in our grant award competition. Okay, that is going to be supported by 10x Genomics. The second announcement is that we are planning to launch our site for 10x Genomics Visium application as well, that I mentioned earlier as well. We are hoping that we will be achieving this pretty much in next couple of months, but I would say maybe, let's keeping my finger crossed, somewhere in month of April, we'll start with the new financial year of India. The third one is that we are soon going to be do on-site hands-on training of for 10x Genomic services as well, right? So we are planning to do host about somewhere around, once or twice a quarter training for students or PIs or industry people. We are really, really going to take you down to the entire process one person at a time. It's not going to be a big group. So please keep in touch. And we are going to announce that through our social media sites as well. So that's another thing, that we are going to give you hands-on training for the 10x Genomic services. Finally, so along with that, we are currently going to offer 20% discount for all the participants. So whoever is participated for this particular talk, congratulations, we have your data set. With that, we are going to provide you 20% discount on the data, which is more than 100 GB on our Noasic platform. We are also allowing 5% discount for all the participants for a single PO. That means everybody gets a discount on the sequencing services, regardless of whatever the service they are planning to use, whether it is bulk or single cell. And if you are lucky enough, you can club multiple things together as well. There is no hashtag or asterisks here in that. It's very open, straightforward, and we can work it out for the discounts with you as well. Finally, this is a very recent one, is that we have recently touched 100 S4 flow cell sequencing on Noasic 6000. Prima's team was here with us to celebrate this. I'm not sure the actual number in general of S4 the world has utilized, but certainly I think we are the first one in India. Probably less than 15 sites from few of my colleagues who are running the centers like this, who are my friends and colleagues I've talked about. Even Cleveland Clinic has not reached 100 S4 flow cell, right? So my alma mater has not reached that capacity. So I'm very happy that we could achieve that mark, being the part of Neuberg Supratech and Neuberg Center for Genomic Medicine. And finally, we are opening a training institute as well in the genomics based training. This is going to be open for master students as well as the medical students as well. And we are going to do an informal launching of that pretty soon. And we are going to call it as a SBRIs. Basically, our site is present on an SBR road, and that's what we are naming it after, right? So that's with that, I think I'm gonna end, and we really hope to host you soon at our Ahmedabad facility, and hopefully see you soon. If not, probably somewhere around next year in the middle of the year or at the end of the year in a user group meeting. And we would love to talk to you as much as possible. With this, I think that's the end of my talk, and we can really take the questions if somebody has. I'm going to stop my slides.
Thank you, Dr. Vishal, for this wonderful presentation. Very insightful.
Thank you.
And at the same time, I congratulate you one more time for becoming a certified service provider, and also for the congratulations for the 100 S4 cells. I'm hoping sooner but not later, Supratech would be running, put another milestone for running 100 chromium chips. So that would be great. Encourage the scientists and the students to take advantage of the grant that Dr. Vishal spoke about. And it's a wonderful opportunity. The grants, the training, and the 20% discount. I mean, take advantage of these wonderful deals. And yeah, about the questions, if there are a few questions, and I'll speak them. Anybody is free to answer. Yvonne or Dr. Avid or Dr. Vishal or me myself, anybody can answer these questions. So there was this first question which Dr. Yvonne had answered in the Q&A box itself. It was about rare autosomal recessive disease, and Dr. Vishal also touched upon it in the presentation itself. So I just want to add that uh, there we already have a paper on cystic fibrosis, which is like one of the autosomal recessive disease. And this research in this particular paper, this paper was published in Nature Medicine in May 21. So you can look for, see, one of the things is the 10x Genomics website. It has a wonderful, place for finding all the presentations which have used the 10x Genomics technology. All everything is beautifully curated. So you go to the website, and you can find the list of publication in various categories. You can also select at what category you are particularly interested in. So cystic fibrosis, there is a paper published in Nature Medicine in May 2021. They have this is from Carrero et al. So they used our technology. And as you know, the cystic fibrosis is autosomal recessive disease. Okay, so another question is like, when it comes to single cell ATAC for infectious diseases, how can one see the percentage of cells with infection? So anybody is free to answer.
I guess I'm gonna give a quick take of mine based on the question. I think that's coming from Shweta, right? So Shweta, I guess I would really think in a way that if you are starting for this particular infection for the very first time without any literature, the quick answer is no, you wouldn't be able to identify whether the cells are infected or not. But if there is some literature available, right, that this particular infection leads to this particular gene expression increase or cell type increase, right, or a particular pathway increase, that's the time you will be able to identify, right? Because now you have a single cell gene expression assay done, or probably you can do even an immune repertoire assay, right? You can identify different cell types along with the B cell and T cell as well, along with the transcript information. And now that you have this information, you would really be able to, based on the transcript information and the clonotype which is present on this cell surface, you will be able to identify that this particular cell was infected with this particular infectious agent, or right? I mean, I think that's where you are pointing it out to. And I think that's when it will really helps you out. So again, if it is for the very first time, congratulations for thinking in that direction. But a sweet answer for the first time would be no, but you would be the first one to do that. So you can really establish something, this kind of a disease-based profile. And India is a hub for the infectious disease as well. So you are not wrong in thinking in that direction. And you can really be brave in starting anything which is for the very first time as well. We are here to help you out for that.
Yeah. Another thing just like to add is that so like Yvonne was showing you, Dr. Yvonne, she was showing you in the slides. So you get to see the clusters in the TSNE plot. So you're talking about like rough sort of percentage at how many cells have infection. See, the thing is, the more the cell, the bigger the cluster. Okay, just as simple as that. And if you have very few rare cell, then you'll probably see a very, very small sort of cluster on the TSNE plot. That will give you an idea. Okay, so moving on, then another question is why can't we study gene expression of immune cells by 3' gene expression? One of the users has asked this question. Anyone can answer, with Yvonne or Dr. Vishal. So my take on this would be that we have a specially designed kit, 5' gene expression, as with Dr. Yvonne and Dr. Vishal, they all discussed in the slides. So the 5' gene expression is best suited for infectious diseases and immune cells because it basically covers all the VDJ regions. Okay, so there is a lot of diversity, and when it comes to immune profiling, and most of it is in the 5' region. Okay, that's why we have a special 5' kit, and with the help of that, you can do the VDJ profiling, and with the help of feature barcodes, you can also do complete TCR and BCR. So that's the thing. So another question. So in terms of organs.
Sorry, Sachin ji. I just want to add to that is that it's that the way to query the cell surface markers, right? So those reagents are not available with the 3' UTR, and I think it's about the technology development. I don't think so. It should be that difficult. It's rather 10x will be coming up pretty soon, but that's the way it is designed as of now. That if you want to query, it is better in the 5'. They're providing with the 5' kit, and simply the reagents which captures the cell surface markers are not available in the 3' UTI kit.
Yeah, right, Dr. Vishal. Thanks. So then another question we have is in terms of what different organs can be studied with the help of fixed RNA profiling or the flex kit. So internally, I think we have covered, we have providing probes that cover, internally we have studied most of the organs, whether it be kidney or the pancreas or brain. So I would say practically all of the organs have been covered in our internal studies. Anything you would like to comment on this, Avid or Yvonne?
So I think, Sachin, right, the question is about the different expression patterns of the same gene in different organs, if they can be, associated to the same protein product from the same transcript.
Yeah.
So I think you're exactly right. We have data sets, for the standard gene expression kit, but also getting that for the gene expression flex kit. I think if you combine this with protein expression, as in if you're looking at specifically, I guess, cell surface protein, that's probably the only thing you can do with our technology in addition to gene expression. You could sort of marry these two outputs together to really be sure what they are derived from. Otherwise, it basically comes down to analyzing your organs separately and really comparing those expression patterns and potentially also using some public data sets that are available from whole genome sequencing or, bulk RNA sequencing studies, I'd say.
Yeah, thanks. So one last question I have. So how do we deal? This came from a user. How do we deal with big size cells? The size which are cells which are very larger in size.
Yeah, Sachin, I can answer that question. This is the beauty of TenX genomics technology, especially because we work with microfluidic channels. So here we can accommodate up to 100 micron theoretically, right? So this is where TenX is market leader. Why? Because other competitive technologies, competitions, they have well-based, micro well-based. Their maximum, the well size is 40 to 45 micron. Right, they can accommodate up to 20 micron confidently. So, but if you see, especially neurons and some cells which are above 70 micron size, right? So when you are loading your PBMCs or some complex tissue cells, so sometimes you will miss out the large cells. So this is where TenX technology is far better, and we have supporting publications where intentionally customers loaded larger size cells and they can able to still identify the different cell types.
Great, Amit, thanks. And that is why we also have the, like Yvonne discussed, and we also have a nuclei isolation kit available. And you can also, if the cell is too big in size, you can also, instead of loading the whole cell, you can isolate the nuclei and work with the nucleus itself. And nuclei isolation kit is also compatible with our FRP or fixed RNA profiling or the flex workflow as well. So that's the beauty of the entire workflow. So another question, suppose after, so this is from Rajesh Kumar Maurya, suppose after WES and WGS, rare sample is extensively reviewed in case of undiagnosed diseases, what is the yield of your technology? So I think the user is trying to ask that whether the rare sample or rare cells, they can be discovered by by the TenX Genomics technology.
So I'll take on that. So I think when you're saying, I guess, Rajesh, when you're trying to say about whole exome sequencing and whole genome sequencing of a rare sample, right? So you're trying to say that you have a lot of variant of unknown significance, and you have a lot of heterogeneity in the DNA itself, right? Or you have a lot of mutations or a lot of large scale deletion, insertion, inversion, things like that, right? And that has been extensively reviewed by the WGSs and WESs, right? So now that once you have this information, and I believe this is coming out from the blood tissue, or rather I'm taking an example as a blood, right? So then you can work out, right, with the PBMCs, we can perform a 3' gene expression analysis. We can identify those regions based on the transcriptomics, right? And at the same time, in this particular set of samples, when you're trying to work it, I think FRP is the best solution, right? Because it is going to increase the sensitivity as well, right? I think Yvonne showed that there was a significant increase. I mean, roughly what I have seen from the publication, I still have to download the data to analyze it by myself, but it looks like 1.5 to 2% increase in the significance of calling of the transcripts per se, right? If those regions are QC'd at the transcript level, right, you would be able to make a correlation based on the WGS data set. A lot of VUSs now can have a role by single cell technologies, right? Because imagine a lab's burden, right? To characterize 10 VUSs is pretty much some scientist's entire career, right? But single cell technologies, specifically in a rare disease scenario, I think we are at the luck that with 10x Genomics is here, right? Because you just need probably 4 or 5 patients and you do single cell transcriptomics on those guys, right? And then you have a quick correlation. And next thing you will so see that in which particular cell lines or what kind of a mouse model we need to generate this VUSs, right? To further characterize it, right? So that's the beauty and power of this technology. So of course it feels like whether you now, if your question is, if you're going to identify a specific cell types with different WES, I think 10x just discontinued those kits. Probably they should be coming up with pretty soon, because that if the probe technology is working, I really hope and think that's my wish to Yvonne and 10x team as well. A single cell exome is the next thing, right? Isn't it? I mean, that's the next level world that we are thinking of. We are not going to identify only VUSs. We are going to identify VUSs in particular cell type. We'll have a precision medicine based on that, right? So that's what we are trying to go through that. And I think your question leads us to that kind of a question, right? So of course, rare disease is the best scenario that you utilize 10x now. You identify those transcripts, you QC that, you have this database, because without database and previous knowledge, we are not moving with the science, right? I mean, my first class of PhD, I'm sorry if I bored somebody, but that's the way we learn science, right? By ruling out the negative results, we learn about the positives, right? We always assume what is the X, and then if X is this, then we prove our hypothesis. And if X is wrong, that at least our hypothesis is wrong, at least one statement is done at a time. And single cell can have answers up to 10,000 hypotheses per time, right? I mean, that's the beauty of single cell, right? So it's about how to think about your experiment. And that's when, again, talk to us. Let us talk to you. Let us train you. Let us explain you what does this data mean. A lot of publications have a lot of underwritten information as well. We can help you, guide you, and you can come out with multiple different kind of an experiments. And I think, I really feel the reason we jumped onto the 10x Genomics as a CSP site is that we really want that India needs to come up on a single cell scenario pretty soon, rather than late. Whole genome sequencing is being taken by multiple different countries, and we will need a support. Everybody needs a support. It's a team effort to reach that scale. And we would really request even government bodies to look into that kind of a profiling of the population in general. So yeah, I mean, if you do that, I guess, I know my answer was a little lengthy, but I think that's the scenario even I dream of, right? That you have VOS identified based on the transcriptome profile. Because if you think about it, in the central dogma of life, that's what you go with, right? You identify mutations in DNA, then you look for that RNA, and then the protein function. When you're talking about rare disease, you have phenotypes and you have a mutation. You are just missing the RNA part of it. And 10x is the best way to go about it now.
That was, I think, very helpful, explanation, Dr. Vishal. And I just want to add, coming to rare cells, that's where 10x has launched HT chip, high throughput chip. Using that tech chip, you can able to load up to 20,000 cells in one single well. And even the same sample, if you can load on the instrument, you can, screen up to 1 million cells. So that is the capacity. So especially example, I can tell you MRD, minimal residue disease, right? Just load your cancer patient sample on HT assay, and you can see whether the cancerous cells are there or not. I'm just saying, no, I think people should work on that aspect so that, they can identify if there are 100 cells also we can identify out of, so that is the beauty now that is available with 10x. So this is the right time for anyone to explore with 10x Genomics, and welcome to single cell revolution. Like that's why in my talk also I said it's a 21st century, it's a century of biology.
Yeah, thank you, Arvid and Vishal, for your answers. I wanted to add that point as well, that we only need a very few cells to actually identify, like to be identified as a cluster. You are able to zoom into that particular small cell cluster, identify the transcripts, and you will be able to identify these rare cells. So that's actually really exciting, even if you just work with 10,000 cells per sample. If the high throughput capability is not accessible, you can still use our gene expression flex kit. You just allocate, 3 or 4 barcodes to the same sample to basically make it 40,000 cells, for example, for your one sample, to just enrich the number of cells that you screen and then be able to detect the few rare cell populations that are in there, as just another way of thinking about running it.
Yes, exactly, Ivan. So that's what FRP is helping. Like if you can't afford HT, so FRP with different barcodes, you can able to pool same sample in different wells. Yeah.
Yeah. Thank you all for the beautiful answers. So I don't see any more questions. So I think we have reached the end of this event, and I'd like to thank certain people, Nishtha, for arranging this, making all the arrangements. Thanks for that. And I'd like to thank Dr. Avid, all our 3 speakers, starting Dr. Avid and Dr. Yvonne for staying this late and delivering beautiful talks. And at the end, thank you, Dr. Vishal, and congratulations once again for being CSP. Yeah.
Thank you guys. Thank you so much.
Yeah. And in the last, I'd like to thank all the audience who have attended this event. And I just want to reinforce, this thing that Supratech is now a certified service provider. So I'll encourage you one more time to, write projects, whether it be students doing their PhDs or scientists doing their project, working at different research institutes. So I'll encourage you to write projects, and whatever be the number of samples or cells you are working with, our CSP Supratech is there to help you. We are also there at Premas are there to help you, and our 10x Genomics team here in India, they have their presence in India as well. Avid is there, and we have FAS from 10x as well here in India. We are all here to help you. So thank you all.
Thank you, everyone.
Thank you.
Thank you so much.
And thank you, Sachin, for taking us through this event today.
Yes.
Okay, thank you.
Thank you.
Thanks.
Bye-bye. Bye-bye all.
Bye.
Bye-bye. Good night, everybody.